Good morning, and thank you for joining the New York Botanical Garden for WeDigBio 2020. My name is Charles Zimmerman, and I'm so pleased to welcome folks from near and far who've come today to learn about biodiversity collections and how we can all make a difference to support their preservation and usage for environmental research and conservation. Over the next 4 days, thousands of individuals around the world are coming together to join in this shared mission. And today we're thrilled to present this series of virtual events to kick off WeDigBio 2020 at NYBG. At any time during today's webinar, please feel welcome to submit questions by clicking the Q&A button at the bottom of your Zoom window or send comments using the chat. For accessibility, live captions can be enabled by clicking the CC button. This presentation is being recorded and will be shared for future viewers in the NYBG Lecture Library. Now, please join me in welcoming Assistant Director Nicole Tarnowski, who's here to lead us on a virtual tour of the New York Botanical Gardens William and Linda Steere Herbarium.
Hello, welcome to the herbarium. Hi, welcome to the herbarium. I'm going to give you a bit of a behind-the-scenes tour of our collections. I'll go through how we store them and create them and what they're used for in scientific research. Our herbarium is the second largest collection of plant specimens in the world. We have 7.8 million specimens. And to give you a sense of scale, the room that you see here is just one of 4 rooms in this building that are filled with herbarium cabinets that store all of our specimens. You can see what they look like, what the cabinets look like inside. This is how we store all of our specimens. We try to keep things very organized because we have so many specimens. They are organized by scientific name, by the family, the genus, and the species. And they're also organized by what part of the world the plant was growing in when it was collected. So you can see everything is stored in these folders and then filed into these cabinets. The cabinets themselves keep the specimens safe by preventing UV light from degrading any of the colors. It also keeps insects from eating the specimens. There's several types of beetles that love to eat dried plants, and so we try to prevent them from having any access to our specimens so that they can be preserved here forever, really. I'll show you in a minute some of our specimens that are actually pretty old. This collection spans plants that have been collected in every part of the world, every type of plant you can think of, and over a very long period of time, close to 300 years. One of the things about a herbarium specimen is that once it's pressed and dried and attached to a sheet of paper, that specimen can exist and be useful scientifically for hundreds, maybe thousands of years going into the future. So let me tell you a little bit more about how we collect these specimens. This is a plant press. So this piece of equipment is very simple. It's basically the same way that botanists have been collecting plant specimens for well over 300 years. It's just a wooden frame with layers of absorbent blotting paper and corrugated cardboard, and then these straps to tighten everything up. If you've ever pressed a flower in a book, you've basically made a herbarium specimen. That's exactly how we preserve our specimens as well. So as a botanist goes out into the field, they will come across something that's interesting to them, and they'll clip off a portion of it with the idea that every herbarium sheet is only 11 by 17 inches. And so they will only clip off enough of the plant that will fit on a herbarium sheet, but they will keep in mind what are the most important structures that you would need to see on that plant. You can see with this particular specimen, there are flowers and there are also the leaves, and the collector was very careful to press this so that you can see both sides of the leaves. and see, once this is glued down to paper and you only see one side of the specimen, you'll be able to see all the important structures. So this is put into the newspaper. It's then flattened into the press. You can see there's all these layers. You can keep adding more and more specimens as the day goes on. This plant press could be 3 feet high if you've had a very busy day. And then the whole thing is put on its side over a low heat source so that the corrugations in the cardboard allow the heat to distribute throughout the entire press and dry the specimen as quickly as possible. As the water's coming out of it, you keep cinching the belts tighter and tighter so that the specimen is as flat as possible. Once you take it out of the press, this is what it would look like. Everything is pressed in the field wherever it was collected, and then they are packed up in the mail and sent to us. So this is what it looks like when we receive it from the field. The other thing that is as important as the specimen itself is the label that tells you all the information about the specimen. So, and Charlie's going to go through in much more detail with you about the label, but every label is going to tell you the scientific name, the family, genus, and species. It's going to tell you exactly where this specimen was collected, hopefully with geo-coordinates so you can pinpoint it on a map. It's going to tell you who collected it, and it's going to tell you exactly when it was collected. So all those pieces of information give you a much more complete picture of this plant, and those pieces of information become really useful data that scientists can use for many different types of research. The final step in this preservation process is taking it out of this newspaper and actually gluing it onto a herbarium sheet. We use something similar to Elmer's. It's a water-based white glue. So we'll glue everything down. And then often the specimen needs a little bit of extra reinforcement with a needle and thread. So there's this beautiful art to pressing these specimens in addition to them being useful scientifically. Another way of preserving specimens, there are some types of plants that don't really lend themselves easily to pressing and attaching to paper. So occasionally, when it's something that's really bulky, like this prickly pear cactus, we will preserve them in alcohol. In the herbarium, we lovingly call these pickles. So as I mentioned, once these plants have been pressed and dried and housed safely in the herbarium, they will potentially be here for hundreds of years. Botanists have been collecting plant specimens for well over 300 years. And we have some of these as sort of artifacts of those very early years of botanists just starting to study plants and document the natural world around them. All of these are, these older specimens obviously were initially collected for scientific data, but because they're so old, they have very interesting histories attached to them these days. One of our oldest specimens in the herbarium was collected in 1769 by Joseph Banks and Daniel Solander. They were the two botanists that traveled with Captain Cook around the world. This was a British expedition, and in their colonial times, going around the world documenting plants and animals. Another specimen that is really interesting. We have so many different stories we can tell about different botanists. This specimen was actually collected by John Audubon, who most people are more familiar with for his work in creating beautiful field guides for birds. But if you think about it, every one of his beautiful paintings always included plants. And so it makes sense that he actually collected herbarium specimens as well. This particular specimen was collected on one of his last trips out west, where he was actually working on a book of quadrupeds of North America. Another pretty famous scientist is George Washington Carver. We have quite a few specimens that Carver collected and contributed to our herbarium. Most people are more familiar with his research on agricultural plants. He was also an excellent mycologist, so he studied fungi. And he had a great working relationship with a mycologist up here in the north named Job Ellis. And so Carver would collect, specimens that were of interest and then send them to Ellis. They collaborated quite a bit. This particular specimen was a brand new species that Carver collected. And when Ellis published it, he named it after Carver. So you can see the genus is Metasphaeria and the species name is Carveri to honor George Washington Carver for discovering this new species. A little closer to home, we have these specimens that were collected in what was known as Elgin Garden. So this was the first public botanic garden in the country. And if any of you that are listening are from New York City, Elgin Garden, the footprint of Elgin Garden is where Rockefeller Center stands today. This garden was founded by Dr. David Hosack. So he was a physician, a practicing medical doctor. And in those times, part of your medical school was to learn the healing properties of plants. And so I think many early botanists actually started out as physicians, and their love of plants just expanded from there. And that was the case with David Hosack. He grew up in New York and did some of his studies back in Scotland and sort of learned sort of traditional botany there. So he came back and started this garden in New York City. He was able to run it with just his own money for about 15 years before he just couldn't afford it anymore. So he ceded the property to the city, who gave it to Columbia College, which became Columbia University. Columbia University held on to it for a number of years. So as I said, this was Rockefeller Center in Midtown now. And so for many years, that's where Columbia College's campus was. And in the meantime is when the grid of New York City kind of sprouted up around this garden, and it became very valuable property. So in the 1870s, Columbia decided to sell the property, and it gave them enough money to move their campus to where it is now on the Upper West Side. In the meantime, there were many other botanists associated with Columbia College, including Hosack, that in the next generations. The founders of the New York Botanical Garden, Nathaniel Lord Britton and his wife Elizabeth, were also associated with Columbia. Nathaniel Lord Britton was a botany professor at Columbia. And really the establishment of the New York Botanical Garden sort of fully realized Hosack's dream of establishing a preeminent botanic garden in the United States. So the story goes that the Britons on their honeymoon, well, actually, let me back up for a minute. This specimen is one of my favorites that they collected together. So they were both part of the Torrey Botanical Club, which is still in existence today. It's associated with Columbia University and with the New York Botanical Garden. And the Torrey Botanical Club would go on field trips. So these are pictures from our Leicester Mertz library of the Botanical Club taking the train up here to the Bronx, right? And so the Britons would go on many of these field trips, and this moss collection was collected by both of them. You can see, because every single specimen includes who collected it, you can see the initials of Elizabeth Knight Britton and Nathaniel Lord Britton. This was actually collected 2 years before they got married. So you can just imagine the sweet dates they went on that were actually field trips of the Torrey Botanical Club. So when they went on their honeymoon, they went to London to visit Kew Gardens, which is another excellent botanical garden. And they were so impressed with the facilities there, they realized that the United States needed something with these scientific facilities here. And so they came back and largely to Elizabeth Britton's credit, convinced the Torrey Botanical Club to put out a public appeal to establish a garden here. And so we were incorporated in 1891, and that sort of brought full circle Hosak's dream of having this great botanical garden here. If you ever are in Rockefeller Center, there is a plaque and that commemorates where the garden used to be. So that's kind of some of the fun historical aspect of what our specimens can tell us. But the main use of herbarium specimens is how they are used by our scientists. So as I mentioned, the information on every single specimen includes what it is and where it grew. And those are the two main pieces of information that botanists need to study a particular group of plants. So someone could be a specialist when you get your doctorate in botany. Often you focus in on a particular plant group and that becomes your expertise. The type of study that would come from studying a particular plant group for years and years would be a monograph, right? So this is a monograph of the pine family or the genus Pinus. So a monograph is going to tell you very detailed description of every single species. It's going to give you beautiful illustrations that are detailing those descriptions. And occasionally it's going to, you can map all of where this species occurs. So you can see on this particular map, every one of these dots represents a herbarium specimen of where the species was collected. And if you have enough collections of any one species, you can start to get a sense of the whole distribution of where that species occurs. So this is one focus of one way of trying to study botany is focusing on a particular plant group. This is just a really cool pine cone. This is Pinus coulteri. It's one of the heaviest pine cones that is known. It's also called the widowmaker. If this drops down from the tree, you don't want to be standing underneath it. It's very heavy, and you can see these little barbs on the edges of each little cone. They're pretty sharp. So that's one way of focusing on the information we have on these specimens. Another way, as I said, is geographical, right? So if someone can be a specialist in a particular plant group, someone alternatively could be a specialist in a particular plant, a particular geographical area. So instead of studying one plant and all the different places it grows, they study one place and all the different plants that grow in that one place. And so the type of publication that would come out of this type of study is called a flora, right? So this is two of our researchers that have spent their entire careers documenting the flora of the Intermountain region of the Southwest US. So this is a massive region of high elevation deserts, starting with the Rockies in Colorado, all the way to the Sierra Mountains in California. It has taken them 40 years to document every species growing in this region, but they published their last volume just a few years ago. So that's another use of our herbarium specimens. More recently, we have been using herbarium specimens more and more for conservation work. Because every single herbarium specimen has a date on it, we essentially have this record of what's been growing on Earth over time and how that has changed. So one type of study that documents climate change is phenology studies. Every single specimen is usually collected either with fruits or flowers. So we basically have documentation of when a particular species was flowering and exactly what time of year. So in a phenology study like this, we have many, many specimens of the same species, all collected in exactly the same place, and there's this other dimension of time. So this first specimen was collected in 1891 and was flowering on June 16th, right? You can see the flowers here. This next specimen, 90 years later, collected in the same exact place, same exact species, was flowering much earlier on May 31st. And then just, 30 years later, this same exact species collected in exactly the same place was collected even earlier on May 25th. So that gives you a sense of, that there's physical documentation of how the natural world is changing over time. And that all is associated with the specimens that we have in our collection here. Of course, we also have representations of very rare plants and things that are starting to disappear. This is a specimen of American chestnut. So this specimen was collected in 1891 here in the Bronx in Van Cortlandt Park. Just a few years before the chestnut blight was discovered. The chestnut blight was a fungus that it nearly wiped out all of our American chestnut population. It was first discovered actually right across the street here at the Bronx Zoo. And this specimen exists that, 20 years later didn't exist anymore, growing in Van Cortlandt. So that's another interesting, really important use of our specimens, is documenting that something used to grow here where it no longer does. These two specimens are unfortunately representations of species that are now extinct on Earth. This specimen was collected in Hawaii in 1911. Of course, a place like Hawaii has an especially sensitive climate, and is very sensitive to changing that environment. And so island ecology can be really damaged very quickly because it's such a small area. As things are changing, there's often nowhere for those plants to continue growing because it's a small region to begin with. This other species used to grow in the Galapagos Islands, which I'm sure many people are familiar with from Darwin's work. We can also think in terms of the landscape changing about invasive species. Obviously, as people move around the planet more and more, we are bringing plants with us that used to only grow in one spot and bringing them somewhere else. And often that means when they grow in a new environment, they become invasive, and they might crowd out the plants that were there before them. If any of you are familiar with purple loosestrife, Lythrum salicaria, you see this growing quite often in sort of wet areas, often even just alongside highways and roadsides. It's very pretty. It's got these beautiful purple spiked flowers, but it's highly invasive and it crowds out other plants that grow in the wetlands. This particular specimen is considered one of the first herbarium collections documenting that this had started growing in the wild. So this plant was collected in 1831 in Massachusetts, and it's sort of considered ground zero for how this plant got established in North America. Now, the herbarium has hundreds and hundreds of collections of Lythrum salicaria that have since 1831 been collected throughout all of North America. So if you looked at all of our specimens and plotted them on a map, you could get a sense of how this plant started moving to different parts of the continent. So that's another really interesting use of our herbarium specimens. We're also continually documenting new invasive plants that are continuing. It's not just plants that got established 150 years ago. There are constantly new plants moving around the world and getting established in new places. This is an invasive that was just documented growing here in the Bronx River just a few years ago in 2014. Previously it came from Asia, and just in the last few years has started to get established. And one of our researchers was able to document that in herbarium specimens that this is now growing here. So, studying some of our older invasive species might help us to learn ways to eradicate and prevent these newer invasives from spreading. Other uses of herbarium specimens can be helpful in studying wild progenitors of food crops, right? So these are two species of wheat, right? So this species is the species of wheat that we currently grow. Obviously, this was cultivated for, you know, hundreds, thousands of years, and started off with, you know, a wild progenitor of wheat, which was the same genus, Triticum. But this is another species of Triticum. And you can see how different the inflorescence and the seeds are. So that's another really important use of herbarium specimens, and is having these related species of crops that could perhaps be used in molecular work to develop more hardy crops as climates change. So we could possibly develop crops that are more water hardy, that are more heat resistant, and are able to survive as climates change. We also we can study plants for medicinal uses. This is a plant in the blueberry family that was studied for its chemistry, right? Its biochemistry. So this particular species was studied for the antioxidants that are found in this particular species. And so that's another interesting use of our specimens, is being able to have these physical specimens to analyze the chemistry of the plant. So that's sort of an overview of the herbarium. I am happy to take any questions people may have.
Thanks, Nicole. That was really fantastic. We have several questions coming in. First one is has to do with the oldest specimen in the collection. You mentioned Captain Cook is pretty old. Do we have anything older?
We do. So Captain Cook was from, what did I say, 1769. I think our oldest specimen is from the 1690s. I didn't bring that one out. We want to handle many of these very carefully. That one I think is in our fungal collection. And I should say, obviously the herbarium wasn't established until 1891, but we have many specimens older than our own institution because we trade them with other herbaria that are older than us. So the Captain Cook specimen, for example, we received from the Natural History Museum in London, who has most of the Cook expedition specimens.
Yeah, we had another question about that as well. Do institutions trade specimens with each other? So it's not only going out and collecting plants, it's also collaboration between institutions and between collectors and determiners. Is that right?
That's right. So anytime that someone collects a plant, they usually collect several duplicates, actually. So that allows us to keep one duplicate here at New York, and it allows us to have additional duplicates that we can share with other institutions. So that helps our own collection, and it helps collections at other institutions as well. And often, you know, as I said, everyone is a specialist in a particular plant. So if I know that there is a blueberry specialist, for example, at Kew Gardens, and I have some specimens that I've collected, I might earmark one of those duplicates to go to the institution where the specialist is.
Great, thanks, Nicole. Someone asked a question about the Amazon rainforest and worrying that, you know, as it continues to disappear, do we think there are a lot of specimens in that area that are still undocumented?
There are. I mean, even throughout the world, there are hundreds of brand new species discovered every single year. And the Amazon is one of those incredibly diverse places that scientists are racing to document as much as they can before things completely disappear. And obviously with the fires, the devastating fires that are continuing to happen, several of our own researchers work very closely with Brazilian botanists in the Amazon to go and document the species growing there.
Mm-hmm. Someone asks, do scientists ever go back and remove pieces of the plant specimen to sample for chemical and DNA studies and things like that?
Absolutely. We have very strict protocols about when that's allowed and when that's not allowed, but we do allow researchers to what we call destructively sample. So they are allowed to take a small portion of a specimen off of the sheet for either DNA extraction or chemical analysis. But we have very strict protocols to keep track of what has happened to that specimen, and also to keep track of what happens to those samples and make sure that those are used properly.
And only a small proportion of the plant can be sampled at a time, right? We can't take an entire herbarium specimen. Everything needs to be done kind of on a micro level.
It is, and we only allow someone to sample any particular specimen once. So if someone 5 years ago had already taken a leaf off of this specimen and sequenced its DNA, there would be a note on the specimen that says that's already been done, and we would not allow a second person to then remove anything else from that specimen.
That's wonderful, thank you. Someone asked, just for a refresher, what was the name of that newly discovered invasive species, I believe in New York or near New York?
Yeah, it's called Corydalis incisa. So this originally was from Northern Asia. It initially got established in North America maybe 20 years ago, but much further south than us. And it's slowly starting to creep up the coast. And so this was one of the first cases of it now existing in New York.
And I think the common name is incised fumort, is that right?
That's right.
Yeah. What was the title and the authors of the flora book from that you mentioned earlier?
So this is the Intermountain Flora, and this was written by two of our emeritus scientists, Pat and Noel Holmgren.
Great, thank you. We've got so many great questions coming in, so thanks for answering it all. We have a question from Priscilla. Do we have any specimens from the Alexander von Humboldt expeditions?
We have a handful. We do have just a few. Mm-hmm. And we have even more of his partner, Bonpland. The two of them traveled throughout South America quite a bit. But we have specimens of each of them.
Great. And you've mentioned that we have, some invasive plants that are present in the herbarium, but there's so many, right? It's not just one or two. It's quite a large number, unfortunately. Yeah,
there are invasives in every corner of the world, basically. As I said, as people move around the world, so do the plants. Sometimes that's on purpose. If the chestnut blight, for example, was a fungus that originally came over here because Japanese chestnuts were being cultivated here. And so that fungus could exist on Japanese chestnuts without completely killing them. But when they moved and started cultivating those Japanese chestnuts here, that fungus was able to jump and really decimate our American chestnut. So that's one example of how things can move. Other times it's more unintentional. The purple loosestrife was probably just seeds that came over in the ballasts of ships. Right, so this purple loosestrife specimen was collected in New Bedford, Massachusetts. That was a shipping town right on the coast, and in all likelihood, that plant caught a ride on that boat, and the seeds were released in the ballast from the ship. So there's many ways that plants move around the world, but there are invasives in every corner of the world at this point. And we have many of those documented here in the herbarium.
It's unfortunate, but at least we know, right, more by the information we have. So I have another question about where does this all get funded by? What supports the New York Botanical Garden Herbarium? How do we keep doing this work in the future?
That's a great question. Obviously, the New York Botanical Garden is nonprofit. The garden itself is extremely supportive of the herbarium and all of our science programs. But we do also search for external funding. We apply for many grants to our own federal government through the National Science Foundation. We're also extremely fortunate to get grants from private foundations. We've had grants from Google. We've had grants from many foundations. So, you know, we have to be a little creative sometimes in getting extra help, but there are many sources out there that are interested in funding the continuation of our activities.
And I'll add that a big mission of a lot of those fundings and projects has been to digitize collections. So bringing the information that's stored on these specimens, bringing that to the digital sphere and allowing that to be shared with scientists around the world. And that's also a big part of what WeDigBio is all about, bringing citizen scientists into the fold and making it so that we're all doing our part to better understand the biodiversity around us. So it's really cool to see what's happening behind the scenes, but also to know that we can all do our own things to contribute. So thank you, Nicole.
Great.
Do we have any more questions? All right.
Okay, well, thanks for listening. This was fun.
Yes. So hold on 1 second. Go back to our other screen. All right. So thank you. And again, thank you everyone for attending today with our virtual tour of the herbarium. WeDigBio has only just begun, and we really look forward to seeing many of you this afternoon as we focus on contributing to citizen science projects that help unlock the collections we learned about today. And you can follow this link here shown on this page to learn more and register for all these events if you haven't already. Don't forget that WeDigBio 2020 lasts from now until Sunday, October 18th. And we hope that you'll take time before then to get involved and participate in all the virtual projects we have ongoing. Your help makes a tremendous difference for scientists around the world, and we're so grateful for your enthusiasm and support. So until next time, take care, and we look forward to seeing you all again soon. Bye-bye, everyone.